Anyway, so I asked Chris Edwards to give a little talk about the physics of upwelling relationship to biology, to kind of talk about the scales and the issues involved with hydrodynamics. So if we're talking about enhanced upwelling or upwelling plumes, there's a component of that obviously that's physics and how long water masses stick together, how organisms respond to that. And Chris is one of the few physical oceanographers who's been willing to talk to us microbial ecologists. In fact, I would say he's now a card-carrying microbial ecologist. So I'll let him take over.
Thanks. As long as the card isn't, there are no tests, I'll be all right on the microbial side. So John asked me to talk a little bit about physical controls, and I'll take to heart what he said this morning, that we're not exactly constrained by the target. What I did was I did my best to put together about 3 or 4 topics that might be useful for discussion here today, but I was doing my best to think of aspects of the system that would be fertile for discussion. So hopefully we can raise some discussion either in the middle of the talk, which would be fine while some material is up here, or at the end as we've been doing this morning. Either way is really just fine. So I'll talk mostly about 3 things, maybe 4. I'll start with physical controls on upwelling in the coastal upwelling system. And I'll talk both about the circulation itself and also about nutrient fluxes that are associated with it. I'll talk some about spatial scales of variability and time scales that are critical. I think the dispersion discussion lends some support for that. I'll talk about calculations that we've done, not for ocean ecosystem processes, but really for understanding marine connectivity up and down the coast. But I think they relate to the discussions that are going on today. And then I'll end with more ecosystem topics related to biodiversity that actually fit nicely into some of the discussions that we had this morning. I'm a physical oceanographer, but I consider myself a more general ocean modeler, and I work with a variety of models to simulate the ocean circulation. Most of the work is with the regional ocean modeling system, which many of you are familiar with. It's a commonly used coastal ocean modeling system and has a lot of nice features in it that allow simulation of both the physical circulation and components of ecosystems and particles, for example, in constrained regional environments. This is just an example of one of the models that we work with. It shows very clearly the region that's my area of focus. The coastline is shown here. I didn't put states in here or rivers, so it's kind of hard to see on this projection. But basically, this is mid-Baja, California, Point Conception, Monterey Bay, San Francisco Bay, Cape Mendocino, Cape Blanco, Columbia River, and on up to the Canadian border. And this represents the domain over which we have very good atmospheric forcing for running models of this type. I'll be talking about aspects of this model as well as more simplified or coarser resolution model output, but that's the context that I'm thinking about the circulation in. Let me just run that again. This particular movie shows sea surface temperature in the simulation off the coast. And if you can see it, it's a spin-up problem. So right in the beginning, there's some adjustment to initial conditions. But you can see it very quickly devolves into a series of eddies and submesoscale features that are really quite mesmerizing. And I think this is one of the things that is useful in thinking about the plumes and blooms and filaments that exist off the coast here. So this is sea surface temperature, with blue being cold and warm red being warm. And you can see a clear seasonal cycle that's taking place with seasonal warming over the California current system and then followed by wintertime conditions. You can see the clear evidence of the coastal upwelling taking place along our coast and the presence of a number of mesoscale features having length scales like 100, 200, 250 kilometers offshore. And for those in the front, maybe if you look at this carefully, you can also see the breakdown of those mesoscale features at the edges, which are the submesoscale processes, which may also be relevant for some of the things that are being talked about today. So the filaments are happening on very small scales, like 10-kilometer scales, and then there are also these 10-kilometer breakdowns of the much larger eddies that exist and populate the California current system. Francisco said some of this, but one of the points that I'd like to make is the seasonality of upwelling on the west coast here. This is true of all Eastern boundary upwelling systems, is that they have a seasonality associated with atmospheric forcing. And here are some figures which help to illustrate that. Francisco showed the development of the North Pacific High. Of course, that's a summertime phenomenon. In the wintertime, the atmospheric pressure system is such that there's what's referred to as an Aleutian low out over the tail end of the Aleutian chain. And that low pressure system has geostrophic-like wind patterns that bring the jet stream hitting into the coast off of the northern part of the US, southern Canada. That Aleutian low gives way in spring and summertime to this North Pacific High as heating changes over the ocean and over land adjust the atmospheric fields. And that's why we get this seasonality in wind stress, in equatorward wind stress. Just to make that point more clear, I show here climatological averages from the month of January and the month of June off of the coast here. You can see the mean winds. This comes from the COADS data set. The mean winds are intersecting the coast relatively perpendicularly up off of Oregon, and we even have downwelling favorable winds off of Washington. Off of the central California coast, we have upwelling favorable winds, but the amplitude is quite weak, actually. So the color scale on here is the amplitude, and you can see it's at the low end of this color bar. Whereas in summertime, as this North Pacific High sets up, we really have intense upwelling, particularly off the north-central part of the central California coast. And this has been well known for quite a long time. This is a nice paper by Ted Strube now, 25 years ago or so, but it illustrates from data some of the variability in upwelling, both in the mean and in its variance ellipses, showing mean downward circulation or wind stress off of the Washington coast with mean upwelling favorable circulation off the central California coast, whose amplitude is largest right here in the region north of San Francisco, and then tapering off as we go down towards the Southern California bight. And this helps to explain some of those biomes that Francisco was talking about. There really are different regions of the California current. The Southern California bight has very different wind stress patterns than the Central California region, and the Pacific Northwest, of course, as well. So it was the seasonality, I guess, that I thought was useful to talk about. We have a very intense upwelling period, but it's of limited duration. And so to some extent, maybe discussions of upwelling enhancement might relevantly focus not just on enhancing it during active upwelling, but maybe in periods when upwelling is not so strong. Of course, then you have issues of potential light limitation and other problems.
Chris,
yeah.
The Southern Hemisphere doesn't seem to have the same seasonality in the high pressure variability.
Very good, thanks. Also, to highlight that variability, I show two upwelling indices. This is from off of Oregon and off of Big Sur here, and the scales of these are different. So the upwelling index, so this represents a theoretical prediction based on atmospheric winds of what the volume transport is within the upwelling cell. It's the well-known back in the upwelling index, and this is produced by NOAA ERD down in Pacific Grove regularly from atmospheric calculations through the Navy. And the point is that you have a very large downward favorable wind stress off of Oregon or Washington at some times of the year, very upwelling favorable at other times of the year, whereas off the Big Sur coast you have a very strong upwelling favorable structure, although there's a very large seasonal cycle in that as well. The other reason to put this up is that this helps to give a volume transport estimate. I haven't seen any of the engineering designs that were talked about in Hawaii, but I don't know what volume we're talking about. And if we're thinking about enhanced upwelling, the type of volume structure we're thinking about is like 200 cubic meters per second per 100 meters of coastline that the natural system produces. So I don't know if these pipes and tubes that are being talked about are larger than that or smaller than that or the same order. Yeah.
The one that Mackay Engineering and Lockheed were talking about was 33 feet in diameter. This is going to be an interesting and righteous experience. The one they were talking about for us was originally 6 feet in diameter, and then they ratcheted it up to 15 feet in diameter. I'm not a fan of wider. They are, because you get less in the way of friction losses. But if we foul our nest, if we bring up too much cold water too quickly, then we'll have a mixing issue with the warm water. So we're listening to everyone on this.
So anyway, this is just a nice scale to think about, 100 cubic meters per second per 100 meters of coastline during active upwelling.
But it also shows we all look at the simple line of upwelling and non-upwelling, when in reality the week-to-week variability is phenomenal.
Yes, in addition, that's a great point, Ken. The variability on even a few-day time scale is enormous, and you can have periods of very intense upwelling followed by weakened winds, which, as we were talking about earlier, can be favorable to growth. In fact, that variability can be a positive impact as well.
That's also different in the coastal corridors, it has to be steady.
Yeah.
There's much less variability?
Yeah, because there's no weather, you're at lower latitude.
Yes, right, very good. The impact of the upwelling favorable wind stress, of course, is to cause upwelling, which is illustrated by these figures here from a different paper by Ted Struven 25 years ago, which shows the isotherms and isopycnals coming up along the shelf off of, I think, Newport, Oregon, from hydrographic experiments. And I think the point that I wanted to make here is what's the depth of upwelling things are coming from, maybe between 100 and 300 meters, depending on the circumstances. And to some extent, that depth plays very well with the large-scale oceanography, which is illustrated in this annual mean depth from Argo floats of the 26.6 density surface. That density surface in this figure is, let's see, 26.6 is right here in this figure down at about 250 meters. That's kind of consistent with this one, which shows a shoaling of the deep layers as we approach the eastern boundary. And this, of course, is part of the large subtropical gyre circulation that Francisco talked about, where over the majority of the subtropical gyre, particularly in the west, but over large swaths, you have very, very deep regions of high nutrient concentration. The large-scale circulation of the subtropical gyre makes the eastern boundaries kind of primed for this upwelling effect. All right, the next topic I wanted to talk about were physical controls on the source depth of upwelling, and this, of course, matters in terms of nitrate fluxes. And the starting point for this is a paper by Steve Lentz and Dave Chapman from 2004, where they were motivated by observations in cross-shore transport from different coastal upwelling zones. They found off Northwest Africa, well, let's see, here are 4 different zones: Northwest Africa, Northern California, Oregon, and Peru. And dots that go to the right of the line are offshore Ekman transport, and dots that go to the left of the line are the return flow coming in underneath that offshore Ekman transport. And these are, I believe, annual average mooring observations. So what they noticed was, of course, in all of the upwelling zones, there's offshore Ekman transport taking place. But if you look in the different regions, you have onshore transport either very deep in the water column for a coastal system down at, say, 70 meters or 60 meters, whereas in some other regions, for example, off Oregon, it's happening immediately beneath the Ekman layer, or let's say more predominantly beneath the Ekman layer and not so deep in the water column. And to some extent, these vary by region, but their goal was to understand what controls that. Did you have a question?
So what makes mass balance off of California? The other ones look like they balance that way.
Well, you're right. So that's a great question. To some extent, you have to imagine mooring observations in a more complex three-dimensional circulation pattern. So they are simplifying, and you're right, there is no mass balance in this particular shot. but there is variability in the depth at which that cross-shore transport happens.
But is that reflecting some important difference in the circulation?
Well,
in these
they would
water
argue, and I would argue also that what this is actually reflecting is this change in the parameter S, which is what's referred to as a Berger number, and it represents a physical change across these systems. So Berger number, in this case in coastal systems, is defined as the product of two numbers and divided by 1, where alpha is the topographic slope, which helps to define a length scale over which variability occurs. N is the buoyancy frequency, and F is the Coriolis parameter, which varies with latitude. So the point is that they can quantify these regions in terms of this Berger number, and you can develop a theory that determines the ratio of transport happening either shallow in the water column or deep against the bottom boundary layer through this parameter S. And that's what's shown in this expression down below. It's the key result, and it's a very simple expression that can be illustrated basically by these figures, or figures that I'll show on the next slide. That for small Berger number, you can have onshore transport that's happening largely along the bottom boundary. Whereas as the Berger number increases, you end up getting interior transport dominating over the bottom boundary layer. And that has a direct impact on the nutrient flux, which is work that we did to extend this work to a more ecosystem type problem. So this is kind of showing the same thing, but now the color map is not cross-shore velocity, but it's a nutrient that exists in the water column, and we divide the domain into a few different regions. So there's a surface mixed layer, which is just the Ekman layer. There's a bottom boundary layer, which is basically this pipe going along the bottom, bringing deep nutrients up towards the surface. There's an interior between those two, and then of course there's an inner shelf region where the bottom and surface boundary layers mix. And the point that I'd really like to drive home is that this Berger number matters in terms of nutrient flux. So when the conditions are such that most of the transport is happening along the bottom, nutrients are drawn from deeper depths. Because of the natural gradient in the presence of a nutricline, drawing from deeper depths generally results in higher nutrient transport into the euphotic zone. So for small Berger numbers, you generally have higher nutrient flux into the euphotic zone. In contrast, as this Berger number increases, you end up reducing the bottom boundary layer transport, increasing the interior transport, which effectively results in a shallower onshore flow and smaller flux to the surface. You can quantify this both in terms of nitrate flux or nutrient flux, or a depth of upwelling. It doesn't really matter, source depth. And anyway, this I think is a useful physical control to think about. There are different regions of the world which have different properties. So we estimated the nutrient flux associated with different, this is work with Mike Jaycox, who's a graduate student at UCSC. We estimated the contribution of the bottom boundary and interior flow to nutrient flux to the surface using climatological profiles from the different regions, and found some differences, for example, off of Peru, relatively similar contributions, both by the interior field and by the bottom boundary. But in contrast, Northern California and Northwest Africa showed very substantial enhancement to the nitrate flux by the bottom boundary layer. To some extent, this is driven by the vertical profiles of nutrients, and Francisco earlier was showing the high nitrate concentration in the Peru upwelling system, and to some extent, it's then driven by these physical controls, the physical properties in the different regions. A second thing which was talked about earlier was also the wind stress curl, and this is a different mechanism by which you can have upwelling taking place. I was mostly referring to the coastal boundary layer upwelling, which occurs over what's called a deformation scale of the coast, but you can also have localized upwelling due to the horizontal structure of the wind stress. And this has been noted in a series of papers by various people over the years, and Francisco mentioned it also, that this Ekman pumping-driven upwelling can drive a substantial portion of the nutrient flux as well. So we looked at that with fairly idealized profiles where we considered no curl to the wind stress structure and curl with varying horizontal length scales. And I think the important take-home message of this is that this, again, alters the degree to which you have interior transport and bottom boundary layer transport. And for us, we actually argue that the no curl case is the one that optimizes net nutrient flux from deep waters up to the photic zone. This sort of result, I would say this is being debated, but this type of result is consistent with other numerical calculations that have been made by other people with slightly different questions in mind. But it doesn't address the important point that has been made that upwelling-driven nutrient flux, Ekman pumping-driven nutrient flux can drive different ecosystems offshore as the coastal upwelling divergence process happening near shore driving the diatom blooms. So people have speculated that the large-scale curl structure to the wind stress is driving picoplankton-supported communities offshore, and that's still an important point. All right. The next topic I wanted to mention was dispersion. And this one I put in because I don't really understand how the upwelling enhancement experiments are going to run. I think of these parcels of water that are brought to the surface and are quickly spread out by the fluid dynamics of the coastal circulation. And so I don't really understand whether you're talking about pumping fluid up and somehow keeping it near the surface, or enclosing it in some way and allowing it to move with the circulation. If it's fixed in one place, I can see how, say, primary production can be enhanced in that location, but I don't see how subsequent trophic levels are enabled to occur in those controlled zones. So I put this up simply because I think of the fluid as a very complex three-dimensional circulation pattern with a lot of turbulence in it. And what we're doing in this animation is seeding the model of the ocean with a number of floats that are transported by the ocean circulation. The different colors represent different depths, black being the surface, going down red, blue, green is finally at 100 meters depth. And there is some variation in the black and the green, although mostly you see the spreading out by these mesoscale features or the plumes that come off the coast, particularly associated with promontories like Cape Mendocino and Point Reyes. So I think of the ocean as these boluses that get brought up and dispersed rather dramatically from their source waters. And I don't know quite how that fits in with the paradigm that you're thinking about. We try to quantify how far these things can go by looking at, these are called Lagrangian probability density functions, which illustrate in a statistical way where things go if they're released from a particular location. So here we're thinking about a release location along the central California coast, say in the Gulf of the Farallones, and asking by season of release where they go. And on average, for example, in wintertime, after 30 days in the water column, they spread out down to Point Conception and maybe up to Mendocino, although mostly they stay pretty locally. In springtime, that down coast transport is enhanced. In fall, because of the change in the wind stress, actually things stay a little bit closer to the coast. You can look at a longshore population connectivity by quantifying these alongshore distances for potential recruitment. And these are other statistical measures of that which show at different time scales you get different dispersion up and down the coastline, and that promontories, capes like Mendocino and Point Reyes, have important impacts on the actual transport of the organisms, at least in the model. We find breaks at these coastal features.
These are point source dispersion of probability plots. You just fed it only from Monterey or?
Well, we actually feed it from all over, but
okay.
In order to create these, we isolate those that were released from some zone in the vicinity of the Gulf of the Farallones.
Yeah.
Okay. And then bin average their position so many days after release.
Yeah, one thing in terms of what Francisco was presenting today was commenting about the shadow of his upwelling. So that he sees the strongest fisheries are not at the upwelling itself, but the strongest point of upwelling itself, but in what he calls the shadow of that upwelling. If you were to do a similar exercise than this one with the upwelling of Peru, would you end up having those portions of water moving into what he calls his shadow zone?
I think, so I don't know the answer to that, although actually I know the person who's doing particle trajectories like this in the Peru upwelling system, Jamie Pringle, if any of you know Jamie. But I think what I would argue is that if you think about this as a statistical cloud, almost certainly that statistical cloud would still exist there, although there might be a stronger mean component to it all year round because the variability is not as strong. Would you agree with that?
Yeah, but I mean, you still see, I mean, you have the areas of the tense upwelling and the shadows in this,
Yeah.
right?
On the right-hand side,
Yep.
the connectivity model on the right.
Yep.
Yeah. And you do see something like that on Peru, but it's less, there's less eddy activity and less mesoscale activity.
Right.
Which is what you said.
So I think probably for the timescales that you're thinking about, these are too long, 30-day, 60-day, 120-day. I'm not really sure. So I'm bringing up one other plot. I'll just jump to this one here, which is from literally our backyard here. This is the Central California coast from the Big Sur region up to about Point Arena. And if you look at dispersion of these clouds, how far do they go? It varies whether you're talking about the cross-shore length scale or the along-shore direction. Things tend to disperse more rapidly in the along-shore direction than cross-shore. And if you let them go out in the water column for 10 to 20 days, in the cross-shore direction, they might go 40 kilometers on average. In the along-shore direction, it might be double that. So there are ways of using these models to quantify these distances, which might be useful for thinking about pens that might get moved around. I don't know.
Or what do we think in terms of blooms? If we think like Dick Dale has calculated, that we see that most of the blooms will take in the order of 6 to 7 days.
Yes.
Basically, that's the scale that we're looking at.
Right, that's why I put this up.
signature.
Exactly. So I thought this was a more useful time scale than the previous one. So I just wanted to highlight two length scales that are important in the physical oceanography. One of them is one that's very commonly referred to as the internal deformation radius. And this is a nice map of it over the global ocean by Dudley Chilton, which shows its variability over with latitude. It's relatively, it depends inversely on the Coriolis parameter. And so it's relatively small at high latitudes, 20, 30, 40 kilometers. But as you go to low latitudes in the vicinity of the equator, the system scale becomes much larger, and you get these eddy scales growing as low latitudes are reached. So if you're thinking about seeding experiments in the tropical Pacific, these scales may be useful. They define these eddy scales and the scales over which isopycnals vary. This global map doesn't help so much in our coastal ocean, so here's an estimate of this length scale off of the Central California coast, and it's showing kind of a 20-kilometer length scale or less as we go in towards shore.
What are the colors on that? Is that the Rossby radius?
Sorry, I'm having trouble going back. Let's see. Oh, the colors on the map? Yeah. There. Yes, this is the internal Rossby radius calculated in the same way as the
Are
Chilton.
the blue ones, blue colors, the
5.
It actually, by this definition, which is a reasonable approximation to this length scale, as the depth shallows, the deformation scale decreases as well. So in the vicinity of the coast, you might have a 5 or 10 kilometer scale. As you move offshore out in the open ocean, it stabilizes at around 20 or 30 kilometers.
But, so that's different than the area of active upwelling. The area of active upwelling, according to Smith, you know, and others, the Rossby radius determines the area where there's actually vertical transport. And
Agreed.
my calculations of this area are that it's about 20 to 30
Yeah,
kilometers.
that's the same as, I would say that's the same as this. So the upwelling is happening off the shelf, which is in this red area here, and that's the scale that you get. But as you come near shore, there's a narrowing of that, which is also reflected in the variability that you see both in Monterey Bay and off of Point Reyes. You know, in fact, this scale, the parcels experience some average of these scales, so I don't think it's inconsistent.
No, I was trying to, I'm sure it is, I'm just trying to understand what you're telling us were in relation to this area, which shows up pretty clearly in our sections, you know, that the area of active upwelling off of California is on that order of 20 to 30. It's a very narrow area. It
is, but it's governed by this, this
yeah.
length scale.
And then it increases as you get closer to the equator.
Yes, agreed.
And
Consistent with this.
Peru, it would be, in northern Peru, it would be 100 kilometers, 100 some kilometers. Yeah.
Right, agreed. The other length scale that's worth mentioning is this sub-mesoscale length scale. I don't do very much work in this particular area, but I thought I'd highlight a few studies of people who do. This is a modeling result from Jim McWilliams' group at UCLA, which shows the changing fluid dynamical structure as you move towards higher and higher resolution and resolve not just the eddies themselves, but also the breakdown of the eddies at their edges associated with frontal processes right at their edges. So this is a 12-kilometer model, 6-kilometer, 1.5, and 0.75 kilometer model. And although there's a general agreement in the structure of this temperature field, in fact, you see a rather more complicated filamentous structure occurring when you can resolve those scales, at least in a model. People have done estimates with ecosystem models of what the impact of this is, and that's what's shown in this paper here by Mahadavan, where she models a frontal system and shows that you get both new production immediately in the vicinity of that front and a straining field which tends to draw out the phytoplankton that has been produced previously. So both in terms of stimulating production and in terms of straining those fields out, this smaller kind of 10-kilometer length scale may be important to think about. All right, the last topic that I wanted to talk about was some ecosystem modeling work that we have underway in our group. This is an ecosystem model that uses what we refer to as the self-assembling ecosystem. Sometimes people refer to it as Darwin. And it's shown schematically up here where it's a typical NPZ type model, except that there's substantial diversity of the phytoplankton pool. Whereas many ecosystem models have 1 or 2 phytoplankton components, we have 80, and those 80 are subdivided into functional groups whose properties differ basically in terms of their nutrient utilization, both in terms of the nutrients that they use specifically and in terms of the rate processes that control their growth, and that's what's shown here. I think well, let's see. Here, I'll show this. Here's an example of one phytoplankton organism that's represented in this model. And I'm really showing it to show not only the structure that's happening out occasionally in the open ocean, but the length scales and to some extent time scales that we at least model production occurring in the nearshore zone. So you can see it's very short time scale, I have to go back again, sorry. Very short time scale with long filaments that extend hundreds of kilometers, 1 to 200 kilometers from shore. Okay. When you sum up the functional groups, you get some representation of biogeography that is similar to what we might expect with, for example, nearshore diatoms dominating this region and picoplankton and other small organisms dominating the offshore zone. There's some temporal variability in the blooms of these things. But what I really thought was useful here for today's discussion was one of diversity. So this represents an animation of biomass of each of the 80 organisms. One of the things that is kind of interesting, we represent 80, but most of them die. Most of them are represented down here. They're maintained numerically, but they don't really contribute to the biomass of the coastal zone. However, about half of them span a number of orders of magnitude in terms of biomass. And you can see by the phasing of these things, they all grow and decay at different times of the year. That's shown to some extent by these color plots up here, but if you just look at it, you find Prochlorococcus, for example, rises out of phase as from the diatoms and out of phase of the non-diatoms. And anyway, that's in terms of timing, that's another issue that it would be worth thinking about. And then finally, this also highlights the fact that only a few organisms contribute to the most substantial fraction of biomass. This is a log plot extending about 7 decades. So it's only the upper few, upper 10, upper 15, depending on how you count it, that are contributing. So our goal was really to understand that biodiversity. How am I doing on time? Am I okay?
You're okay.
Okay. We were motivated by terrestrial and freshwater studies such as these, where people have tried to quantify how biodiversity varies with productivity. And it seems to me, in the ecological literature, which I'm not an expert in, that there is no consensus view on this topic. In fact, you can have diversity increasing with productivity. You can have a number of studies which show diversity decreasing. There are a third of them which show there's no relation to productivity. And then another third which are showing what's referred to as a unimodal relationship. And the unimodal relationship is sometimes argued to be true by figures like this, which is hard to see a unimodal relationship in. It certainly has highest points up here, but it just looks like a big blob to me. So anyway, this is kind of an interesting basic ecological question: is what is the relationship between diversity and productivity? And so we used our model to examine that, and we defined biodiversity to be represented as species richness. So we counted the number of individuals that contributed to the upper 99.9% of the biomass, and a map of the annual average of that richness field is shown in the left panel here. Richness was largest in the oligotrophic portions of the gyre, where growth is suboptimal, and a number of organisms are competing for the resources. The species richness declined as we moved in towards the coast, as the upwelling produced a more intense upwelling bloom, and it also decreased along with greater growth rates up towards the higher latitudes in our domain. And just for reference, if we add productivity to that, you can see all of the productivity is largely happening near shore. So we created our own blob, which looks like this, and I think it similarly looks like a relatively unconstrained scatter of points, where productivity is shown on the x-axis and species richness is shown on the y. If you show this on a log plot, it goes down at the other end, but I'm just showing it on a linear plot, which shows high species richness at relatively low productivity. And that species richness declining as we move towards higher and higher levels of production. And I put this into this talk because I thought it would relate to this question of seeding populations, and whether, in fact, if you have this enhanced upwelling, if you can maintain a monoculture or a small number of organisms, at least our modeling study would show that under the highest productivity levels, you end up with very few species that are thriving the most.
I've always been taught that the standard biodiversity productivity relationship is exactly the one that you're. So more productive systems are less biodiverse. And Bill Peterson up of Oregon shows that when you have an El Nino year, copepod diversity goes way up because you get these offshore waters coming to shore.
So this is consistent with that.
Yes.
Yes. So I think
Which always leads me to ask the biodiversity people if they think that more biodiversity is actually better.
Excellent question. So we can look at that in the model. And that's what this figure shows, which is asking the question, do you maximize productivity by having a monoculture? And these results are preliminary, but they seem to suggest that you don't maximize productivity by having a monoculture. So there may be some value in thinking about seeding populations not with one organism, but with a couple that might be able to thrive. Now, it may not be possible to make that conclusion from our study, which averages over various biomes within the California current. And so I'm really talking about maximizing productivity over a very large range with possibly different environmental issues. But it certainly suggests that a monoculture may not be what you want. Where basically, if we look at a polyculture of, say, 10 organisms in the CCS, we get a certain productivity. And as we, one by one, in a random way, decrease the number of organisms that are present in that system down to 1, we get a substantial drop-off in total productivity. So if the goal is to both increase upwelling and maximize that primary productivity, you may want not just one, but a few organisms that can compete and survive in this particular environment that you do it in.
The paradox of the plankton. Who wrote that?
So, okay, that's the end of my talk. What I was trying to do was just present some ideas that might be useful for the topics that are under discussion. I mentioned the substantial spatial and temporal variability in atmospheric forcing that occurs along with the amplitude of upwelling that the natural system produces. I talked about a theory which discusses the depth of upwelling and the associated nutrient fluxes that we think are physically controlling at least that first step in the process. I discussed both the deformation scale, this Rossby radius, and the sub-mesoscale, smaller length scale that are important to think about when designing one of these experiments, and finished off with this discussion of biodiversity that hopefully helps to lead the discussion in terms of this seeding. So that's all. Thank you. Yeah, Jim?
We've talked a lot about upwelling. We haven't talked about relaxation of upwelling. Can you tell us what you've learned from the models about the importance of these cycles of upwelling and then relaxation, the alternation of the surface transport?
I should have a better answer for that. The fact is we mostly look at it in statistical averages and not episodically. But you know, what I can say from a non-technical analysis is that the upwelling takes place and the production occurs no matter where it is. That is, the production takes place either near shore if there's a relaxation event, or offshore if there's transport offshore still with intense upwelling. I think, and we were talking about this a little bit this morning, I think that the mixed layer depth plays a role in the speed of that upwelling process, where if you have a deeper mixed layer, the average light intensity is somewhat lower, and the speed of primary production is reduced a little bit. But it's not as though we have a substantial amount in our models of nutrients that ultimately don't get used. So there's a question of time scale, there's a question of location where it happens. My sense is that with upwelling, you have with a relaxation event, you have things happening more intensely over smaller space scales and faster near shore. But I don't have a nice statistical analysis to actually show that. There's a question here.
Yeah, I just wanted to make sure I understood your point about the biodiversity part. So the way that I sort of think of biodiversity in the phytoplankton is that there's this, when you have low chlorophyll, you have an abundance of these small ecoplankton that are just sort of there. And then if you want to increase the amount of chlorophyll in a system, those guys are still there, but now you have organisms that fill in this next level of chlorophyll. So you have those. As you go up in chlorophyll content, you get bigger and bigger cells, but you haven't gotten rid of that baseline pico group of organisms either. So I was just trying to understand that relative to what you're pointing here.
So we find even in the coastal environments, although they're dominated by our model diatoms, we do find other smaller organisms are there at varying levels. In terms of the biodiversity, our definition of it has to do with the top 99.9% of the biomass. And so the biodiversity decreases by that measure.
Oh, you're saying what the diversity is of the large fraction.
Yes.
Right, so as you get higher up in the chlorophyll,
The others are not counted anymore in
Yeah,
this
so, okay.
measure.
Okay, so that's
But Ginger, I think perhaps the counter is true as well. In the offshore regions where you have the small little things dominating, the big things don't disappear.
right.
They're there. They're just
Exactly.
as they may be just as abundant, but I think you still find more species in the open ocean than you would in inshore, which is counter to exactly what you're thinking, which is you keep adding species as you get to the more productive. I don't think that's correct. I think at least from the biogeography, the people that work on biogeography of other groups as well, basically define these areas of high productivity as much less diverse as in counting the number of species in the open ocean. I don't know that anybody's really done. I mean, now that we have all this genomics,
Yeah, right. Yeah.
you guys may change the picture,
Right.
but
Right.
the standard thinking is that there are more species in the open ocean than there is in the coastal areas. So any textbook on biodiversity,
Yeah.
mainly driven by zooplankton and other things, will suggest that these areas of low productivities not only have less, have most of their biomass in less species, but there's also less species. But open for discussion.
Yeah.
Hans.
Chris, I think Jim's point, the relaxation, that's going to affect the species diversity too, because it's going to affect modal versus sinking organisms.
Definitely. Good point.
So, I didn't want to throw it on the level of.
No, actually, there are, I think, a lot of interesting effects. The other thing, which of course happens during that relaxation, is you can bring offshore species in towards shore, and then when relaxation ends and you have another active upwelling, there's a lateral exchange that takes place. So there's a seeding aspect of that, I think, which is a different mechanism than what you were just describing. So I agree. I didn't mean to say that it wasn't an interesting point with a lot of rich questions to be asked. We haven't looked at it in the way that really has a good answer for it.
Well, we studied that in the Co-op West project off Bodega Bay, and it came up with a couple of different ways what the optimal cycles are for the wind cycle, because basically it all depends on wind. And basically takes maybe 1 to 2 days to spin up, getting nutrients to the surface, and then the best thing to happen after that is to have about 4 days relaxation. So you get approximately a 1-week cycle would be the best for the phytoplankton, and how that matches up with the atmospheric cycle is the important thing that determines the total productivity.
So by your measure, is the best thing for the phytoplankton would be to be near shore. So are you saying that if that fluid was transported offshore, it wouldn't bloom as well?
That's right.
What we found out was very similar, basically, with the wind and the oscillation between upwelling and downwelling. But the other thing that to consider is that the 7-day, if you keep the productivity close to shore, is not only that I agree with you that productivity will keep going if it's offshore, but the same thing that Francisco was saying this morning, you keep less energy for foraging. where you don't have, where if you dilute your bloom because you are advecting it offshore, any organisms that will feed on it will require much more energy to get the feeding. If you keep it closed, you concentrate.
But
your
that's
food.
a second
It's a
trophic
second-
level
it's a
issue,
second level,
which
yes.
I agree with that 100%. It could be that that this effect has an enormous cascading effect on higher trophic levels. But in terms of the primary product production, I think it's happening in both circumstances.
Is there a relation between horizontal mixing and speed of recurrent?
Speed of the current?
Yeah.
Offshore advection.
Yeah.
Well,
because the observations are that in these areas where there is rapid flow offshore, it looks like mixing horizontally exceeds the growth rate in some sense, so you can't accumulate. And I'm wondering if there
yes,
if there is a relation, a well-known physical relationship between, KZ and
this straining field. Yeah, that's an interesting question. I don't know of one. But certainly this dispersion that takes place under high wind conditions, large mesoscale, submesoscale structure, that will dilute these. And so the point is that you may never get some critical mass of phytoplankton that could be grazed upon. So it's not only that it's really this dilution effect. Yeah. Agreed.
Do you have any way of, we sort of talked about this before, and I was wondering about it during the conversation with Hawaii. It's not about the diversity thing, it's more about this question about the bolus of water, kind of where it goes, how long it stays around, and whether it stays around long enough to accumulate anything. So do you who work on such systems have some idea of what, say, the residence time of a column of water is under upwelling versus relaxed conditions?
This is the closest that I get to it, which really shows things move really quickly in the coastal zone. Now there may be times one could imagine constructing adaptive instruments that would, under some circumstances, turn things on when the conditions are just right. But if one just lets it run, my sense is that they move from their source regions quite quickly. Tens of kilometers from their origination zones.
In 10 days, in one
Yeah.
day?
Oh yeah, in 10 days, so these are clouds that disperse. In 10 days, we find 50-kilometer alongshore standard deviation.
But your model has continuous upwelling with no relaxation.
No, this has realistic forcing.
As winds.
This has realistic winds. This is a realistic simulation. So it's not a constant wind stress at all.
Did you look at the latitudinal?
No, I think these statistics, we have looked at this type of latitudinal variation between the Oregon coast, the Central California coast, and the Southern California bight to look at these connectivity problems. But the general story is similar in those zones. The mean transport varies, and maybe the eddy amplitude varies some. But these PDFs, I have them. I could show. They don't look all that different at higher latitude or lower latitude. I don't know off the Peruvian upwelling system.
Yeah, my first reaction would be that they would get smaller and smaller, maybe not by orders of magnitude. So instead of dispersing by 50 kilometers at 10 days, you might disperse at 40 or 30 as you get to lower, as you move. But anyway, if you ever do them, I'd like to see that.
Like I say, I was just last week in touch with someone doing it in your neck of the woods, and I think you'd be interested in sharing those. Yes?
Chris, could you advance a few slides here? The one where you show the band of diatoms along the coast. You're showing the spatial distribution of the, yeah, that one right there. So this figure on the left-hand side is showing this narrow band of where the diatoms are predominating right along the coast. So what are the attributes of the diatoms in the model? That
Great question. So this relates to the discussion we had earlier, where we were talking about whether it was a growth rate process or grazing.
Right.
We have actually both mechanisms in there. We have diatoms have a faster growth rate when conditions are optimal than the other species in the model. And we also have a grazing penalty for diatoms. That's sort of a 20% grazing penalty. We found that the grazing penalty was not a significant contributor to this structure, but that the growth rate was critical. That if we did not have that higher growth rate, we were not able to, in fact, get this biogeography. So
they're the first responders to this pulse.
well, everything responds, but they outgrow everything else that's responding.
What's the scale of that 100 kilometers, 50 kilometers?
Offshore?
Yeah.
Yeah, it's 50 kilometers. It depends, whether you look at it in a log scale sense or a linear sense. Well, anyway, we can talk about those sorts of issues. I won't say that this is a perfect model of the coastal ocean. There are actually a number of flaws to it, but I think it's useful for the types of questions that we're looking at.
Chris,
Yeah.
backing up to Jim's question, we were told first by Mark Hildebrand in June, July when we were down there, and then subsequently, one of the reasons that the diatoms predominate as long as they've got silica is because silica, the silica shell requires, what is it, 20 to 30% the energy of a green alga to build its cell wall?
Yes, so they require the silica source. Is that?
Yeah.
That's right. So when conditions are right, they can really take off, and the upwelling does supply silicate along our coast, just like it supplies nitrate. But there certainly could be regions which could be silicate limited. It so happens we're not finding a silicate limitation, but that certainly could happen.
Another comment that was made by Dr. Benneman is that the silica itself contains some of the energy required, which is not something I understand.
You're asking the wrong person, I'm afraid. But someone else in the room.
If anyone in this room knew the answer, we'll find it out later, but it apparently is one of the reasons that we're all running computers, because silica is so obliging in terms of growing crystals.
Sorry, I don't know much about that chemistry.
Correct me if I'm wrong, but the laying down of the shell is a biological process, which requires ATP energy.
I assume so, but the comment was made that the silica in some way, shape, or form contains some of its own energy that is needed to. It's the reason it grows faster. I don't know the reason